Welcome to the last Chapter 8 lecture for Blackboard. We're going to go through Section 5 and Section 6, talking about dilution, some reactions, and some other properties of solutions. So keep in mind that when you dilute a solution, you're only adding more solvent. So the volume changes and the concentration of the solution is going to change, but the amount of dissolved solute that you have in there is not going to change. So you might have your concentration of solute in grams of solute or moles of solute, but the amount is not going to change. The amount of solute before you dilute it is the same as the amount after. So the amount of solute is told by the concentration of the solution times the volume you're using. So if you wanted to, you could go through a conversion, figure out how much solute you have, and then put that solute into the new solution that you have. But most students find it easier to use a dilution equation. So the last equation shown on this slide, C1V1 equals C2V2, is sort of a shortcut equation that students find helpful for calculating solutions. So let's try an example using that dilution equation. If water is added to 35 milliliters of a 20% sodium chloride solution, enough water to make 120 milliliters of solution, what is the concentration of this new solution? So we're going to use the dilution equation, and here are our initial conditions and our final conditions. At first we have 35 milliliters of a 20% solution, and then we end up with 120 milliliters, and we want to know what the new concentration is. So if we plug these values into our dilution equation, C1V1 equals C2V2, then 20% times 35 is the unknown concentration times 120. And so if we divide both sides by 120 milliliters, again, keep the units with you, so the milliliters will cancel and our new concentration will be in percent, just like our old concentration was. So our new concentration is 5.83%. And this makes sense because we're diluting a 20% solution, so we should have a lower concentration when we stop. Here's another problem that you can use the dilution equation on. So in this case, you want to make a solution of 500 milliliters of a 0.75 molar sodium hydroxide solution. And you have a more concentrated solution, 6 molar solution, and you need to dilute it. And so you want to know how much of your 6 molar solution you're going to need to add water to dilute it to 500 milliliters. So here are our conditions. Our final conditions, we want 500 milliliters of a 0.75 molar solution. And our initial conditions, we don't know the volume, but we know that it is 6.0 molar. So if we put these values into the equation and solve for our volume, the concentrations will cancel, 0.75 molar and 6 molar cancel, and then our answer will be in milliliters. So this makes sense that we need a small volume of our concentrated solution. So if we were making this solution, we would add 63 milliliters to a flask and then add water until we had 500 milliliters, and then we would have our diluted solution, 0.75 molar. So we can convert between the volume of solution that we have and the moles of solute that we have in it by using molarity. So if you use molarity as your concentration, you can convert between volume and moles. And so once we have moles, we can use that to calculate the stoichiometry of different reactions. Here's an example that shows what I mean by that. So you have an equation, you have a balanced equation between nickel chloride and sodium hydroxide. And so you're told how much nickel chloride you have in terms of a solution. You have 75 milliliters of a 0.200 molar nickel chloride solution. And so if you use that much, how many grams of nickel hydroxide will you be able to make? So you are going to need to convert that solution of nickel chloride, the 75 milliliters of 0.2 molar, you're going to use those two numbers to find out how many moles of nickel chloride you have. And then you can use the coefficients in the reaction to convert the moles of nickel chloride to nickel hydroxide. And then you can use the formula mass of nickel hydroxide to convert it to the grams of nickel hydroxide. So here is the equation that you go through. The first step converts the 75 milliliters to liters, and then you multiply it by the concentration of nickel chloride to get moles of nickel chloride. And then you have a 1-to-1 ratio of nickel chloride to nickel hydroxide, so you now have moles of nickel hydroxide. And then nickel hydroxide is 92.71 grams per mole, so you would end up with 1.39 grams of nickel hydroxide as your product from that reaction. So let's try another stoichiometry reaction using solutions. So how many milliliters of a 1.5 molar potassium chloride solution are needed if you're going to react with 300 milliliters of a 0.8 molar lead nitrate solution in the following reaction? So this reaction gives the coefficients that tells a mole-to-mole relationship, and we are given a volume of a lead solution. So we need to use the concentration and volume to figure out the moles of lead nitrate, and then figure out the moles of potassium chloride, and then figure out the volume of potassium chloride solution that we would need. So just as a side note, there is an interesting graph at the end of this section, the end of section 5, page 325 in the text if you have the 4th edition, and that lets you know what sort of things you're going to be using with stoichiometry depending on whether you have a gas or a solution or just a weight of things. So in this case, this is what you would do. You have the 300 milliliters of the lead nitrate solution. Use the second item listed to convert it to liters, and then use the concentration of solution to convert it to the moles of lead nitrate. You're going to multiply it by the moles per liter, and then you'll have moles of lead nitrate. Then you use the coefficients to convert to moles of potassium chloride, because there are 2 moles of potassium chloride per mole of lead nitrate. And then you would divide by the concentration, 1.5 moles per liter of potassium chloride, to get the liters of potassium chloride that you need. And since the problem specifically asks for milliliters, you would need the final conversion factor to convert it to 320 milliliters of your potassium chloride solution needed to do that reaction. So section 6 of the chapter talks about different properties that solutions can have. And the first thing it talks about is the difference between solutions, colloids, and suspensions. Solutions is what we have been talking about so far. They're homogeneous, they're clear, the particles are very small, evenly distributed, they do not settle out. These particles are small enough to pass through semi-permeable membranes. Colloids are also uniformly mixed, but not to the microscopic level. The particles are larger and look cloudy. They can pass through filters, but not through semi-permeable membranes. But they are small enough that they do not settle out. And then suspensions are the heterogeneous mixtures that are not uniformly mixed. You can see chunks or at least cloudiness, and these particles are large enough to not be able to pass through filters. So you can filter these particles out, and or else you can also let them settle. If you wait long enough, they will settle out to the bottom of the mixture. In lab, we also looked at freezing point depression and boiling point elevation, how things that are dissolved in water will lower the freezing point or raise the boiling point. So your textbook also mentions this in that section and points out that it's a property of the number of particles in solution. So if you have a solution of 1 mole of sodium chloride in 1,000 grams of water, that has a larger effect than 1 mole of methanol, CH3OH, in 1,000 grams of water. Because 1 mole of methanol stays together as molecules, so you will have 1 mole of particles. But 1 mole of sodium chloride, when you dissolve it, dissociates into sodium and chloride. So you'll have 2 moles of particles. you'll have 1 mole of sodium and 1 mole of chloride. So the 1 mole of sodium chloride will have twice the effect. So the text also talks about osmotic pressure. And osmosis is when water can diffuse through a semi-permeable membrane that perhaps is keeping larger solute particles from passing through. So if you have concentrations that are different on different sides of a membrane, then it tries to even itself out. So whatever can pass through the membrane will try to pass through. If you have water on both sides of a membrane and water with a large molecule on the other side, and the molecule can't pass through, the water will flow into the side that has that large molecule, trying to dilute it and even out the concentrations. And so that will create some pressure. And your textbook has a picture of some liquid level being raised on one side of a semi-permeable membrane because of that pressure. Most of the time when we talk about osmosis, we're talking about cells, not membranes inside of beakers. And cell membranes are also semi-permeable membranes. They will let water and small particles through, but not all large particles. And so if you have concentrations that are different on different sides of the cell membrane, they will try to even itself out. If they're not, water can flow into or out of the cell, just like it did with the permeable membrane in the beaker. And this can cause the cell to swell up or to shrink, which is usually not what you want. However, if the concentration of particles is equal inside and outside the cell, and it's even, then you won't have a flow of water and the cell won't be disturbed that way. So something with equal concentrations to what you find in a cell is called an isotonic solution. And so a lot of the solutions that we use in medical applications for IVs and other things are isotonic solutions, that they don't cause cells to shrink or burst or anything bad to happen. If you don't have an isotonic solution, you're either low or high, you're not equal. Hypotonic solution has a lower concentration than what you usually find in a cell. And so if you have a lower concentration outside the cell and higher inside the cell, the water will flow in to try to even out the concentrations by diluting the solution inside the cell. And the flow of water into the cell can make it swell or burst. A hypertonic solution has a higher concentration of solute than what you find in a cell. And so if you put a cell into a hypertonic solution, the water that is in the cell will flow out of the cell to try to dilute the outside solution and even it out that way. And that causes the cell to shrivel. And there's a picture of this inside your textbook. The final thing that the chapter talks about is dialysis. It's the use of a semi-permeable membrane to separate solutes. It's a lot like filtration, but since it's a semi-permeable membrane, it can separate particles that are too small for filter paper. And the most common use that we've all heard of is when someone's kidneys are not working properly, they might have to go for dialysis. And so the blood is passed through a filter or passed next to a filter that leaves the larger particles inside the blood, but removes the very small solute, the waste products like urea, from the blood.